Hi everyone. We sincerely apologize for the break. It was due to network issues from our end here, so it ended the stream, and we had to like try to come back online. So we really apologize for that. Please let me know if you can hear me now. Please, can you confirm if you can hear me now? If we are together now? Okay, thank you so much, everyone. Um, please, we're still trying to wait for more people to join. So please kindly just wait as we allow more people to join the call because of the break. So we're trying to just wait to have more people join us. Our facilitator will join us back again shortly. So please just bear with us while we allow more people to join us. In, in case you knew anyone who was here before now that has probably left or did not know that we're back again, you can just let the person know that we are back online. Let them know we are back. Okay, so I guess we'd have to just continue now so that we don't waste much time further. So Miss Effie, please you can please go ahead now.
Okay, thank you so much, Ma. So I guess I will just start all over again. It seems I didn't know where the call the meeting ended. So for everyone's sake, I will just start everything from the beginning so that we can all follow very well. And also, like I said, please, if for any reason you can't see my screen or you are not hearing me, please just feel free to reach out and tell me, okay? So let's get started. Firstly, I firstly said that we are going to start with what we know and move into what we don't know. And what many of us know is the wet lab process. That's the traditional scientific research method. Now, this wet lab process, what is the wet lab process? What do we call wet lab process? These are experiments that are done in a controlled environment in the lab. They are controlled experiments that are performed in the lab. Okay, and this has been the foundation of science in general. Like every groundbreaking knowledge that we have now of on science, whether in biology, in medicine, in chemistry, whichever aspect, wet lab has been the foundation. And we have quite a lot of experiments that were done. We have the Mendeleev pea plants experiments in biology. This was carried out by crossbreeding pea plants in a controlled environment. And why did he do this? To understand the inheritance patterns, right? We have the um, study of the cells and the cell study and all that was carried out mainly with microscope and staining techniques that were performed in the lab. Then in biotechnology, we have the polymerase chain reaction, which is the PCRO. This is a laboratory process that is used to study the DNA samples. So it amplifies the DNA, and this amplified DNA allows researchers to study small DNA samples. Okay, so all these are processes that are carried out in wet lab, and we have um, things that are peculiar to wet lab processes. We have the pipetting. What's pipetting? Is the use of a pipette to, transfer either chemicals or organisms from either the test tube into the um, microscope tray, or wherever we use the pipette to move um, the samples, right? We have the culturing cells. Culturing cells in microbiology, we know about culturing, where you get a particular sample, let's say Escherichia coli, and you put it in a controlled and favorable environment and allow it to multiply. Why? Because you want to study something about it, right? We also have PCR experiments, like I've already explained, is amplifying the DNA to study small amounts of DNA samples. Okay, we also have sequencing experiments. Sequencing experiment is carried out to organize the genes, to put the DNA in an, let's use the word alphabetical and an orderly manner, so that you can understand what you are doing. Okay, after sequencing, after doing everything, it's put in an alphabetical or an orderly, let me use the word orderly manner. We have chemical assays. These are things that are peculiar to wet lab. Chemical assays, this involves the use of chemicals. We know in all wet lab processes, chemicals are used. Okay, and in the wet lab process, after because of the advance in technology and the high throughput methods that have emerged, the high technology that have emerged, the volume of data that are generated from experiments has increased just dramatically. Okay, if we are to use the Mendeleev's pea plant experiments now, he didn't work with up to 10,000 plants. No, I think it was about two or so plants, but a little amount of samples was what he used to get his results. But now, as there has been an increase in technology and advancement in technology, you can work with 1,000 samples at the same time, and all these samples will bring out results. Now, because of these large data that have been generated, especially in the medical and the biological sector, we need computational analysis and bioinformatics to help understand, to help support wet lab processes. Now, this brings us to the dry lab processes. So the dry lab of modern scientific research, it has served as a support system to the wet lab process. Okay, The what is dry lab? Dry labs are scientific research, refers to scientific research conducted using computers and computational techniques rather than the physical laboratory experiment. Okay, I'm going to come again. I says the dry lab research is refers to scientific research conducted using computers and computational techniques. Okay, so in dry lab, with scientists or researchers, they analyze the existing data, existing biological data. This could be the DNA, this could be protein, it could be the genes, but existing biological data, they analyze it using specialized softwares and programming tools. Okay, specialized software. And things that you do with dry lab, we carry out DNA sequence analysis, we carry out genome annotation, we carry out protein structure prediction, microbiome analysis, even evolutionary studies. All this can be done now using dry lab. DNA sequence analysis, like I said, sequencing is putting the DNA in an orderly manner, right? So, and we know the DNA, especially the DNA of humans, is quite large. They extend into gigabytes and terabytes of data, most of this research. So all this cannot be done manually. You need computational assistance, right? So using the computational analysis, computational techniques and computers, we are able to reduce the load. Yes, wet lab processes are still needed, in the sense that wet lab processes gives us the data that we need for dry lab. The because dry lab processes exist now does not mean that wet lab processes are no longer needed, or the wet lab is no longer needed. No, the two now work hand in hand together to give a full and proper research. Okay, the wet lab gives us the data that you need in dry lab. Wet lab, you have the raw samples. So let's say you carry a blood sample, you can't just immediately get the data from that blood samples. No, you need to take it through the wet lab processes of, getting the samples, getting the data. After getting the wet lab, the data from the wet lab, that is where dry lab comes into play. And depending on the research, depending on what you want, your research is about, that will determine the kind of dry lab process you are going to be um, having. Okay, so while wet lab is responsible for generating biological data through experiments, such as sequencing DNA, culturing, and all that, dry lab research, it takes this data and it performs an in-depth analysis. When I say an in-depth analysis, to extract meaningful biological insight. So analysis that you normally wouldn't be able to carry out because of the volume of the data. Because we are using computers and computational techniques, we are able to do this analysis. And dry lab method helps us to also interpret the data. You see, when you get your sequenced data, sequenced genes from sequenced DNA from the wet lab processes, you are going to be seeing a bunch of alphabets, a lot of alphabets, sometimes symbols that you may not really know what it means. So you may just be seeing a lot of A's, T's, C's, G's, just up to millions of those. It looks really confusing to the human eyes. You really don't know what it means. Let's say you're studying a particular genetic disease, you don't even know where that disease is in those bunch of A's and T's and C's you're seeing, right? But because of the computational biology, computational techniques that we use, the dry lab processes that we use, you are able to know, okay, the exact region you are working on, you are able to get the exact protein form from the, you're able to get in-depth knowledge on this data, raw data that you are seeing. So that brings us to what we are talking about, which is bioinformatics. You see, what is bioinformatics? Bioinformatics is the application of computational statistics, of computing statistics, mathematics to analyze biological data especially large data sets generated from modern technologies. Like I said, with the advent of high throughput methods in wet lab processes, we have the new generation sequencing and all that. We begin to get very large amounts of data. We begin to get very large amounts of data from our analysis, from our wet lab processes. And because of all this, we need computational processes. We need computational processes. And it's not just only biology now that is involved. Because you have to use computers, you need computing or computer science. You need to have knowledge on computer science. You need to have knowledge in programming, even statistics, because we know in the normal, if as a researcher, you know that after doing your analysis, there are some calculations and all that are involved. This is the statistical methods that we need. It is also involved in bioinformatics. So all these are what makes up bioinformatics as a whole, okay? And we have major areas of bioinformatics. We have the genomics, which is the study of the whole genome. The whole genome is everything about the organism, the DNA, everything about the organisms, the whole genomic process. Now we have transcriptomics. Transcriptomics is the study of genes expression using RNA data. So if you are conversant with molecular biology, you will know that we have the DNA, we have RNA, and we have proteins. And we know RNAs are gotten from transcribing the DNA, right? It's gotten from transcribing the DNA. So these RNA data, they are studied because when you study the RNA, you're able to know how genes are expressed. Why is that? Is because the RNAs are what carries the message, what carries the code. Though the DNA has the DNA is like the full book. It has the full everything. The DNA is like a book, let's say your full biology textbook, while the RNA is just probably the summary of just a chapter. The summary of chapter 1 is the RNA. It's not the summary of the whole book, but the summary of just chapter 1. That is what the RNA is about. You can see I didn't say it's chapter 1. No. The DNA is and studying the whole book is what we call genomics, okay? You study everything in the book from chapter 1 to whatever chapter it is. Let's say we have 1,000 chapters in that book. Studying that whole book is what we call genomics. While transcriptomics is the summary of chapter 1. So it's the summary of one part of the DNA, just a little part of the DNA. That is what RNA is. So it carries information of just a part of the DNA, and that information will result in protein formation. So when you study this RNA, you know how genes are expressed. Then we have proteomics, is the study of proteins and their functions. Now everything in an organism is as a result. So the reason why somebody is fair is because there's a protein that somebody fair. Why somebody is tall is because there are proteins, combinations of amino acids which forms proteins that causes that person to be tall or short or whatever features you see. So proteins, when you see them, proteins, studying the proteins and their functions is what we call proteomics. Then we have metagenomics. Genomics now is microbial communities in the environment. Okay, we have you know that in every environment there are microorganisms, not just one, but large amount of different microorganisms in the soil, even in the air, in the water, in food, wherever, even in the human body, in animals, in the blood, in the gut of animals, in the gut of humans, in the stomach. there are quite a large community of microorganisms that are surviving there. So when you decide to study, okay, let's say we want to study the microorganisms that are living in a water body and how those microorganisms maybe affect the water environment, maybe the fishes in the water, the organisms that surround that water, how those microorganisms affect them. That is what we call metagenomics. And it's not just studying them at a physical level, is going deeper to study them at a genetic level, studying up to their DNA. You want to know, okay, these microorganisms, how does the DNA of these affect the DNA? You are studying them at a genetic level. Then we have structural bioinformatics. This is studying the structure of the proteins. Remember I told you these proteins come together to give us the phenotype of and organisms, and each of these proteins have different structures. So when you try to study the structure of the proteins, that is structural bioinformatics. Now you may ask, I've heard of what bioinformatics is about and the parts of bioinformatics. Who are the people that can now work or can use bioinformatics or that can get involved in bioinformatics? And we are a whole lot. You have biology, every aspect of biology, plant biology, animal biology. As far as it is biology, you can be involved in bioinformatics. Microbiology is also there. We have genetics, we have biotechnology, medicine, pharmacy, agriculture, computer science, statistics, and mathematics. What you need for you to, all these are people who can work in or can be involved in bioinformatics, that can use bioinformatics. But for you to use bioinformatics, there are skills you need. We said at the beginning that bioinformatics is what you having using computing tools, biology, statistics, and mathematics. Now you might be saying, I'm a microbiologist, I have no knowledge on computer science, I have no knowledge on programming and all that. Can I still work as a bioinformatician? Yes, you can. You just have to learn some of the skills, and the skills that are needed are basic molecular biology knowledge. This is for people who do not have biology knowledge at all. So these are for the mathematicians, for the computer scientists, for maybe even the statistics, the agriculture, even people in agriculture may not have full knowledge of this molecular biology. As far as you do not have knowledge in molecular biology, you can learn. Molecular biology is not something so tedious for you to learn about. You can learn about it. It's something you can easily learn and understand. Then you need to have knowledge on command line and Linux skills. When we use some of the tools that are used in dry lab processes and bioinformatics, all have to do with programming and command and Linux. So you need to have knowledge in them. You may not have to be a guru. This is for people that are in the biological field. You might say, how can I learn about this? Do you know how long it takes to learn programming? No, you don't have to be a guru that you can program or create programs or codes from the scratch. No, you don't have to do that, but you just have to have knowledge in these things, in Python and R. Okay, programming language like Python and R, and some of the tools that we use have been made easy for us as bioinformaticians, that all you need to do is to give it an instruction to do, and it does it for you. But you just need to understand how to navigate through them. That is why we say you need to have knowledge on command line, on Linux, and programming tools. Some experiments need programming tools, programming languages. Some need Linux skills and all of that. So depending on the aspect you are going into, we determine how much of programming and computational skills you will need, but have a knowledge in them. Then you need to understand the biological databases and tools. We have a whole lot of biological databases. We have the NCBI, Enzembo, a whole lot. They are the tools where all this data, remember I said from wet lab processes, we have large amounts of data, and these data could be going in running into gigabytes, terabytes, and they can't be stored on your system. You can't store them on your system because they are too large. It can cause your system to crash. But when we have databases that allows researchers to store these data, and also another reason is so that a researcher in America that has carried out the wet lab processes and have sequenced and gotten this data, when he puts all those data in those databases, somebody in Nigeria here can go to that database, carry the data that have been there, and you are able to carry out your research all on your dry lab. So it's you need to know how to navigate through them. The tools that are used, you need to know how to use them. Okay, there are tutorials that teach you how to use some of these tools depending on what you want to do. You have to have knowledge on this. So after knowing all this, you might be asking, how do I now start? What do I do to start as in bioinformatics? Firstly, you need to take courses, online courses, training program. This is one of the first step into transitioning into as a bioinformatician. Okay, take courses. There are a whole lot of courses out there. Even if you go on YouTube, there are a whole lot of courses. Genomark as a whole also provide courses and tutorials and trainings, workshops, boot camps, and all of that that can help you transition into bioinformatics. Then practice using publicly available data sets. If you remember, I said that scientists, we know in Nigeria, it's not easy to carry out all these wet lab processes. One of that is due to funding. There's not enough funding to carry out all these analysis, wet lab processes, because they are quite expensive. But there are people, especially in America and all that, who have been able to carry out researches like this. They've done the wet lab processes, and they make their data available on all the databases that we have. So you can go there, get these data that have been made available, and you use it to practice. Okay, when you practice it using your computer, carrying out the processes every time, you are able to increase and hone your skills in bioinformatics. Then also participate in workshop and research processes, research projects. Wherever you see a workshop on bioinformatics, on dry lab processes, jump at it. Some of them may not be free, but one thing as a researcher and as a scientist is that you can't get everything for free. Everything may not be available for free, but you know what you are looking for. Bioinformatics is a skill that as a researcher, as a scientist, it is something that you need, and it is beginning to become a projects that these are ways to get started in bioinformatics. And lastly, you may now ask, okay, so as a bioinformatician, what career opportunities do I have? Is there a workplace I can work as a bioinformatician? Yes, there are a lot of career opportunities as a bioinformatician. I've just stated some of them here. You have the academic research, which we all know, lecturing, academic, really, carrying out research academically. You have biotechnological and pharmaceutical institutes, industries. Now, you see, bioinformatics and pharmacology in general is beginning to tilt more into the bioinformatics aspect, because now pharmacologists are beginning to see and experiment on how drugs can be created for individuals based on their genetic makeup. So all these need bioinformatic tools, even biotechnological industries too. Okay, public health and disease genomics. This during the COVID-19 period, there was a call out for molecular biologists, for bioinformaticians, biotechnologists. The reason being is because for you to understand why an organism or why a disease is spreading or is being it's rapid in spread like that, you need to understand the DNA. You need to understand the genes. You need to understand the protein structures. All these things are what molecular biologists and bioinformaticians they do, right? So in public health, in disease genomics, in hospitals, you can work as a bioinformatician. In personalized medicine, like I said initially, pharmaceutical companies are beginning to check how drugs, how treatments can be made for individuals personally based on their genetic makeup. Now, everybody has, no matter how closely related you are, you cannot be the same genetically. You cannot 100% be the same genetically. Okay, there is always a difference, even up to twins that's identical twins that are the same. There are still differences. Okay, so you can't be the same 100% with another individual. Because of that, people, doctors, pharmacists, they are beginning to see how they can create drugs personal to a person. And can they do this? This is through the genetic makeup of an individual. Then lastly, in agriculture and environmental genomics, remember I said one of the aspects of bioinformatics is metagenomics. And this is what? Studying microbial organisms in the environmental level. So when there are disease outbreak in the farm, in where there are waterborne diseases and all of that, understanding how these organisms got there, how they function, what can be used to treat them, all this can be done using bioinformatics. Okay, so agricultural industries also are beginning to see the need, even for plants. You can a lot of plants now are beginning to be to agriculturalists are beginning to understand the need of going into the genetic level to improve the plants. Okay, so either to make it bigger, to make it more resistant to certain disease, to make it more resistant to some environmental conditions, maybe like heat or like cold or like water drought, whatever. They are beginning to see the need and the significance of using the genes to improve these plants. Okay, so all these are career processes career opportunities you have as a bioinformatician. So that is the end. I want to know if anybody has questions for me, and I'll be happy to answer your questions. Thank you so much for your patience and for listening.
Thank you so much, Ms. Sefi, for the beautiful webinar. Thank you so much for the insights that you have shared on how to get started in bioinformatics and dry lab research. And I hope the audience have been able to glean a thing or two from all that has been said so far. So just before she leaves, I would like us to appreciate our speaker. And also, if you have any questions, you can now proceed to ask your questions. Uh, I'll be checking through the comments now. If you have questions, please drop your questions. And also, let's appreciate her if you've learned anything at all. Let's do well to appreciate her. Okay, William Stemida is saying thank you for the session. Thank you for the insight. Andrew is saying thank you very much for the wonderful session. Ustanshia is saying thank you. Thank you so much, everyone, for appreciating our speaker. And I hope it has been worth your while this evening. Thank you for staying up till this moment. And we really apologize for the breakout that we had earlier. There has been a lot of weather disturbance in our side for some time now, so it's kind of affecting the network. But we want to appreciate everyone that stayed through to the end. So thank you to everyone. Zaran is saying thank you. Oluwaseyi is saying thank you. So please, if you have any questions, you still write on the call to attend to any questions that we might have. So if probably there was something she said during the course of our presentation that was not clear, that you need more clarity on, she's still right available here to provide more clarity or guidance that you might need. So please let's quickly do that so we can round up the session so we don't have to keep ourselves waiting on the call. So let's do that as soon as we can. Right, so I am assuming that there are no questions from our end as I am not seeing uh, any questions in the comment section. So I want to believe that our speaker did a very great job. Okay, so I just see a question popping in now. Okay, so this person is asking when Genomark is starting online courses that can help newbies studying biochemistry and wish to go into bioinformatics. Okay, great. Actually, we've been offering a lot of online courses, virtual courses for anyone looking to, begin their journey into bioinformatics. So I don't know if you are a part of our community yet. And by that, I mean if you are in any of our WhatsApp groups. So if you have been there, you would have seen that we roll out, so much opportunities, daily, weekly, monthly. There are always opportunities for everyone. If you are looking to start out or if you're looking to, grow your expertise in bioinformatics and genomics. So also, we also put out these opportunities on our LinkedIn page at Genoma Corp. That's why we ask our audience to follow us on LinkedIn. So in case you miss it on the WhatsApp groups, you can catch it on LinkedIn. So those are like the two major places where you can look out to for some of these opportunities. We have internships that you can do. We have, workshops. We have short-term trainings that you can do to begin your journey into bioinformatics. So what I would advise is that you try to follow us on LinkedIn and always be on the lookout for our posts. Also try to be a part of our community. I believe you must have seen this flyer somewhere or you must have gotten to hear about this program somewhere, so you can ask the person to share you the link of our group so you can be on our groups, because that's like the first place where some of these opportunities get shared to. So yeah, we've been offering courses before now and we look to doing more for everyone so everyone can be a part of this move. Currently there was even a training that is going on for newbies, just like you, that is looking out to start, that is looking out to start your journey into bioinformatics. So there are a lot of programs over here at Genomark that you can actually enroll in. So please just try to be a part of our community, be on the lookout for all these courses or programs that you can take. And I assure you that it would help you even as you are just trying to begin. Okay, so I think we have a question for the speaker. She says, please, can you give us examples of databases that we can get access to raw data? So, Sefi, if you could please speak to that.
Okay, so I initially said there are a lot of databases, but the most common and the easiest for you guys as newbies, I will give you NCBI, National Center for Bioinformatics. Okay, so that is it, NCBI. You can just go on your web browser and type NCBI, NCBI. Okay, it gives you quite a whole lot of data sets. Depends on what you are looking for. You can navigate through it. And like our moderator said, Genomark also has a lot of courses and trainings that helps you in navigating through these tools and this website and these databases. Okay.
All right, thank you so much. I hope the person that asked has taken note of that. So, someone is also asking, please, Mark, can you tell us the duration that it can take in becoming a bioinformatician? I mean, the duration averagely, because I know this is dependent on the person learning.
Okay, so if I understand correctly, you're asking how long it will take for you to stand independently as a bioinformatician. Now, I can't give you a duration because this all depends on where you are learning, how you are learning, and the practicals you are going, how much you are practicing. Okay, so it could take you 6 months, 1 year, 2 years. It all depends, like I said, on where. Where meaning, are you within an organization where you are learning and carrying out bioinformatical analysis? What you are using to learn, if maybe you are using um outsourced tools, free tools, maybe you are watching through YouTube, or do you have a structured plan? Maybe you have a page or a mentor or someone who is putting you through, okay, through all this. If you are just going through YouTube, okay, using YouTube or using seminars and webinars to learn bioinformatics, this might take you a whole lot of time because you have to navigate through where to start and where to end. You know, it's all jumbled up, and you have to first detangle it before you know where to start. Okay. And how much you are practicing. Are you in a position where you are carrying out this bioinformatic analysis every day on a daily basis? All these are what helps you to improve yourself as a bioinformatician. So that is, I can't give you a definite time, but it all depends on your inputs as a person.
Thank you so much for that response, ma'am. Thank you. So we have another question. I think that should be our second to the last question for today. So this person is asking, is it necessary to build portfolio projects as evidence to the use of tools? I don't know if you get that.
Okay, that's if it's necessary to maybe have a portfolio or have projects that you have done.
Yeah, to
Yes,
work as a bioinformatician.
yes, it's nice to have it because when you are asked what have you done in bioinformatics or what tools do you know how to use, it's nice to have things that you can show. But you see, as a bioinformatic analyst, as a researcher, your portfolio isn't necessary. The tool, the project, the work itself, but actually the result. So have you carried out research? So it's what most people want to know is, have you carried out research that involved using these tools? Okay, can we see the result of your research? Has this research undergone publications? So telling them you have you've done I've done a research project, and this research project I had to use this tool and this tool and this tool, and this research has actually been has been published. That is already a portfolio on its own. Okay, so it's not the way you have as maybe a let me use a graphic designer, where you begin to save all the designs you have done, and you're showing them, I made this design, I made this design, I use this tool, I use this tool. No, as a bioinformatician, your portfolio is the fact that you have carried out research, you have done a number of research using some of these tools, and most of these research have been published or on the way to publishing. Okay, that is your portfolio as a bioinformatician.
All right. Thank you so much for your response, ma'am. I hope the person that asked has gotten the clarity that he or she needed. So okay, so Motura is asking, can an undergraduate do this? I think I'll just answer that. Yeah, that yes. Being an undergraduate does not stop you from delving into the field of bioinformatics, and it even gives you a lot of advantage, as you would be starting right early, not waiting until you finish school. So it's going to help you much more if you start now, even as you are an undergraduate. So I think our last question to our speaker for today. I think this person is has a background in statistics and is asking that, as a statistician, how do I fit in? I think into bioinformatics and all that has been discussed so far for today. So the person is asking how he or she fits in to the field.
Okay, if you remember correctly, when I was giving people those that can be involved in bioinformatics, you saw statistics as one of them. So as someone who studies statistics or is into statistics, it doesn't limit you. You can actually go into the field of bioinformatics. Okay, because one of the backbone of bioinformatics is statistics. We carry out statistical analysis. So having a knowledge that is, you see, there are three backbones in bioinformatics: the computational aspect, the biological aspect, and the statistical aspect. So you already have a knowledge in one out of the three. Okay, so you just have to get yourself grounded in the other two, but it doesn't limit you from going into bioinformatics.
All right, thank you so much for your response, ma'am. So I hope the person that asked to has gotten the information that is needed. So thank you so much, everyone. So Adalbi is asking, is there any institute in Abuja where I can be grounded in bioinformatics? Well, I don't really know about that. So maybe if you try and do a search online, maybe you would find one. But as far as Junwak is concerned, we do not have any branch in Abuja. So we are only stationed in Ogbomoso and Lagos for now. So that's where we have, but that does not stop you, because most of our courses here at Genomark are online, they are virtual, and you can take them from anywhere that you are in the world. So it should not stop you from, taking our courses and enrolling with some of the programs that we will run. So yeah, thank you so much, everyone, for joining the session. Thank you for staying till the end. Thank you for being here till now. So we really appreciate our speaker once again for our time and the insight that she has shared. I know that for everyone who joined today, you've been able to take one or two parts, and I said before we even started that the most important thing is that we take action with everything that we have heard today. So yeah, it is with this that we'll be bringing the session to a close, and we hope to be in your faces again soon. So please you can always come back to this video on YouTube. Just search for Genomark Institute, and then you can always come back to this channel to watch the video just in case you missed something. And also you can take time to go through the channel, as we've had so many live sessions in the past that might interest you, and you might also be able to gain a thing or two from. Also, we want to urge you to follow us on LinkedIn, as like I mentioned earlier, that a lot of our programs and courses that we offer have always been rolled out on daily, weekly basis. So ensure that you follow us. And also, if you are in our community, ensure that you don't leave the WhatsApp groups, because you are always, sometimes the first to get to know about some of these updates. So if you're looking to join our WhatsApp groups, so just message anyone that shared the flyer with you, or from wherever you got the flyer or you got the information about this session from. Just send a message to the person, and let them reach out to us so that we can share the link to our groups with you, and then you can also be a part of us. So thank you so much once again. Until some other time that we come your way, do enjoy the rest of your evening. Bye for now.